All right. Hi, everyone. Welcome to this spotlight session of the 2023 Healthy Longevity Global Innovator Summit. I'm George Setfin. I'm an assistant professor of molecular and cellular biology at the University of Arizona, and I received a Catalyst Award in 2021 from the National Academy of Medicine. This award is focused on identifying synergistic longevity interventions. So we're really focused on drug discovery toward increasing healthy lifespan. Uh, if you already have a drug in mind, you always have, if you want to apply that to a human, you generally have to go through preclinical testing in something like a mouse model. And that can be, and that's a very important step for showing that an intervention is effective in mammals. But earlier on, if you really want to do large scale drug discovery for a phenotype like lifespan or late life health, mouse models are simply too expensive to do that at scale and to do these true like high throughput drug screening. So the model that we work in is C. elegans, which are very commonly used short-lived invertebrate model. They live about 3 weeks, and they at least replicate many of the underlying genetic pathways that influence aging. And this model lets us do true high throughput screening directly for phenotypes like lifespan and health span. And when the Catalyst Award came on, we had just finished the initial development of a new screening system to do high throughput lifespan analysis in C. elegans. So for those of you not familiar with working with worms, the typical way of measuring lifespan, as shown on the left, is you sit at a microscope and you take a little piece of platinum wire and you go in and you kind of bop every worm on the head. You do this over and over again to see if they're alive. And you do that every 1 to 3 days over the course of their, the month or 2 months, depending on how long that population is going to live. And this takes a lot of human labor to do these experiments. So if you've got 1,000 worms in your experiment, you're looking at every one of those worms every few days. If you have 10,000, same thing. So we've automated this. We've basically taken a CNC wood cutting platform, so computer controlled wood cutting. We replaced the wood cutting head with essentially what's a microscope. It has a light source, a camera, and it basically moves around, drops down on a plate, takes low frame rate video of the animals. Instead of hitting them on the head with a piece of platinum wire, it flashes a bright blue light at them. They don't like that. They start moving. And so you get both measurement of activity, how much they're moving, and also you can use that information to determine how long they live. And so the Catalyst Award came online and was really the first funding we received to start looking more high throughput at aging interventions. And one other advantage of this particular system, in the normal system, we have these animals in group culture, and when you look away from that plate, you look back, you can't really tell which worm you're looking at. So you really have a population level of longevity and not an individual level. We adopted this system called the Worm Hotel, which was originally developed by Chris Fang Yen at the University of Pennsylvania, which houses animals in basically mini Petri plates on a 240 well grid, and you surround that with a moat of copper sulfate, which the worms don't like, so it kind of makes them stay in their individual little room. And that lets you track these individual animals over the course of lifespan. And by doing this autonomously and using machine learning and image analysis, we kind of take the human bias out of the scoring system. So this system is more robust. It allows you to get single worm activity and lifespan data and do this at high throughput. The current system, we can run about 20,000 worms in parallel, which means we can do about 150,000 a year. And we currently have 2 operating, and we're building a 3rd. So you can kind of see how this is able to scale. And just to give you an idea of what data comes out of this, because we're doing individual worm tracking, we get lifespan, which is, of course, our primary metric. But if you look at this heat map on the right, you also have every row being a single animal, and every square is the activity of that animal over the course of a day. And so you can get a sense for the change in activity of the animals as you get older. And just on the bottom is showing a single activity trace where you have kind of a healthy active period of life that at some point the worms sort of start to become paralyzed, they can't move, and so we call that a metric of health span, and then ultimately we can determine when that worm dies. And we're also working on a bunch of other metrics like body size and velocity and more detailed things that are still in the works. So what do we do with the Catalyst Award? There's really three things this award let us do that's set us up to really start using this system. The first was just defining the metrics of what we need to do, and the first of these was dosing. So initially we had a couple of internal interventions that were working really well by giving the intervention at egg. So when the worms hatch, you just hatch them right onto that drug, and then when you put them on these worm motel plates right at the beginning of adulthood, they get a second dose, and that was enough to extend lifespan. We picked a bunch of interventions from the drug interventions from the literature that had been replicated and shown to work, and this wasn't really working across many of these. So we went back and we revisited our dosing. We started by doing a lifelong dose starting at egg, and then going do 4 additional doses for a total of 5 doses. That worked for a lot of them. A few of them didn't really like that egg dose, like there's something wrong with the development exposure to that drug. And really, if we're looking at aging and thinking about things that we'd eventually want to translate all the way into a human trial, what we're really interested in these is drugs that we can start later in life anyway. And so what we settled on was this 4 dosing protocol starting in adulthood. So you wait till the worms have gotten through development. And this has worked for most of the drugs that we've looked at. This is able to give us a robust and reproducible phenotype that we can move forward with. And so using this protocol, we started by, this is non-systematic, but we did go through and grab a bunch of interventions from the literature and tried to replicate some of what was in the literature. This was partially to validate the system, but also one of the current issues in science and in the aging field is the need to replicate observations, and so this system has let us do that to some extent. A lot of these did actually replicate, so a large fraction of the ones that we studied. I'll note that the ones that didn't replicate, there's a kind of a big subgroup and a notable subgroup, and these are the NAD precursors, and at least in our hands, we've not been able to get those to replicate. But kind of across the board, most of the things that we found in the literature that have been reported are replicating. So that's good news for the field, I think. And we hope to continue to do this at a little larger scale and a little more systematically. We've also been able to collaborate with a few other groups. I'm showing 2 blinded drug screens because these are still in the works. One of them didn't work, that's okay. And another one was fairly successful where most of the compounds from this kind of mechanistic target that we were looking at did work. And so we're now to the point where we've gotten through the validation, we've really got the system working, and we're pushing this into kind of drug screening, usually more in like a targeted mechanistic way. But then internally, we're starting to move to some larger scale drug screening. And the third thing this is done, and what we really want to do ultimately with the system, is not just find individual interventions that extend lifespan, but what happens when you start to combine different drugs? Can we do a lot better? And so we've started taking a few of these drugs that are working really well in our hands, and we've started combining them. We've gotten to maybe 15, I think, combinations at this point. Kind of the first thing I'll say is that the most common thing we're finding, again, that we haven't done this large scale yet, so this is kind of early stage, like preliminary data, is that the most common thing we see is non-additive. So there's 4 examples on the right and a 5th shown blown up. If you just combine these drugs, you don't do better than the longest lived of the individually treated drugs. I noted we have individual activity, so shown on the bottom is the area under the activity curve for individual animals across these populations. And here we start to see maybe some divergence between these, even though we're seeing non-additivity. In some cases, like the one on the left, we do see that the activity kind of replicates the lifespan, but there's other cases where it doesn't. So for the top one on the right, it looks like, or sorry, the second one down on the right, so with the resveratrol rifampicin, in this case, one of the drugs seems to improve activity, but the combination pulls that back down. So we may be able to get some more information as we really dig into these details about what's going on under the surface for some of these. There are a few examples that we found where the combination is not as good as the best of the two individual drugs. And so, for example, caffeine in a couple of cases, while it does give you a smaller lifespan bump on its own, it kind of pulls back the lifespan bump that you get from a couple of other drugs. So here we're seeing that like none of these are bad for you, but maybe the combination is not additionally beneficial and it's maybe a little worse than just going for one of those interventions. And then we're starting. We haven't really hit a home run where we see a big additive effect. There are a couple of examples of that in the literature. And again, we've only done about 15 combinations, but we are seeing a couple of interesting effects. And this drug rifampicin in particular, there's several cases where even though the majority of the lifespan curve is not pushed back, we see this interesting long maximum lifespan tail. And because we have a lot of animals, we can actually do some statistics on that and detect these things. But there may be something about this longest lived population where we're really pushing that in this group, and we're interested to continue that. And we've even done a couple of triple combinations at this point, and again with rifampicin in this case, where with the triple combination, it maybe even push that maximal tail up a little bit. So we may be seeing kind of a new mode of maximal lifespan extension that we'd like to explore more here, but we've just got the early hints. So this is where we're at with the synergistic longevity extensions. We're continuing to do this and build out our profile of combination drugs. But what the Catalyst Award, in addition to some of these collaborations, what it's allowed us to do is basically demonstrate that this system is useful and that we can do high throughput screening. And we were just awarded an NSF grant through the Infrastructure Innovation for Biological Research Award at the Division of Biological Infrastructure. So we got a 3-year, $1.3 million award to basically build the next version of this system. And what we want to do with the next version, right now we're just getting physiological data. We can get lifespan, activity, metrics of maybe body size. But worms, one of the other advantages is that they're transparent and it's very easy to express fluorescent biomarkers. And what we're doing is adding a new multi-channel fluorescent imaging module. So as we go across lifespan at different time points, we can collect multi-channel imaging data, and then we can start with a worm that's expressing 3 or 4 fluorophores that report on different pathways and really gather mechanistic data about what's happening to important pathways that we know are important for aging while we're collecting this physiological data. And this really moves us from kind of high throughput lifespan analysis to high content data collection and aging, where we can collect both the physiology and molecular data at the same time and really start to learn and get more out of these high throughput screens. And as part of this, we're also building a public database. There isn't currently a really good database for storing and analyzing lifespan data or these time series data like we see with activity and fluorescence that we're going to be generating. And so we're going to build that as part of this award. And then to kind of take advantage of this system, most C. elegans transgenic strains express just a single fluorophore. So we're going to build out a panel of multi-fluorophore biomarker strains that are really optimized to use with this system. And so with that, I will go ahead and wrap up and just say thank you to everyone who's been involved, and certainly to the National Academy of Medicine for providing this funding, which really kicked off our high throughput screening system. Thank you.
Thank you so much, George. That was really helpful to hear the overview of your project and how the Catalyst Award helped to take it to the next level and increase the throughput for your research. And congratulations also on the recent National Science Foundation grant. That's amazing. We have about 1 minute left, so I'll just ask you one question. Could you kind of share any guidance that you have for fellow Catalyst awardees that are working to obtain funding following their initial Catalyst Award? It sounds like you've been successful in that, but just general guidance either for folks kind of working in your field or more broadly.
My strategy has been to really just apply for kind of a bunch of different awards that in different areas all related to this one system. So we've probably put in 3 or 4 different applications, and in this case, persistence has paid off. We've at least we're developing a device, and so at least this may be experience of others, but we're developing a device that's for basic science, and we've had a challenge getting interest from places like the NIH maybe to do that. So really identifying a funding source that's really interested in developing kind of tool development for something like this. I think that's what ultimately got us was we've tried a bunch of these awards. We really found one that was interested in doing primary tool development, and now that we had some good, like compelling preliminary data from this award, this Catalyst Award, and some of the drugs collaborations, that I think that really pushed us over the finish line.
No, that's so helpful to hear. And I think links back to something that was discussed yesterday on our securing funding panel about making sure there's a good match between the researchers and the funding entities. So
Yep.
that's a great point. Well, thank you again so much for sharing about your project. And we hope that you are all able to tune into the next session, which will happen just after a quick break. It's the wrap up, future directions, and celebration of our 2023 Catalyst Awardees with NAM President Dr. Victor Zhao. So please do join us for that session as we close out this year's summit. Thank you.